Good afternoon. It's a pleasure to welcome you to the Florence Mahoney Annual Memorial Lecture as a part of the NIH Directors Wednesday Afternoon Lecture Series. For those of you who may not have had the opportunity to know Florence Mahoney, the lecture series began when she was very much alive in 1987 with the Lewis Thomas Lecture, and she was a fantastic woman, a proponent of research, aging research, but the NIH mission in general, who was enormously successful in her salon, in effect, in Georgetown, entertaining presidents and congresspersons, largely responsible for a lot of the growth in NIH and for the existence of NIA. She loved to talk each year before this lecture about who the speaker was going to be, and she was very frank and strong-minded about it, and she wanted it to be rigorous science, none of this wishy-washy stuff. And I think Florence, who was able to express that opinion firsthand up until her death at the age of a very vital 103, would have been absolutely thrilled with today's lecture. It comes at a time when all of us are very familiar with big data and the challenge of big data translating to knowledge, but that translation itself and the understanding of networks that link data has been an area perhaps of some of the most very exciting growth in this past year or two. And we're fortunate enough to have with us today one of the real leaders in that area, Eric Schadt. Eric is here from the Icahn School of Medicine, where he's director of the Institute there of Genomics and Multiscale Biology. He was identified by Thomson Reuters this year as one of the most outstanding, influential scientific brains in the world, and I think it's well justified, not hyperbolic in this case at all. So it's with great pleasure and without delaying the experience any further that I introduce him to you. Eric?
So many thanks for the invitation to speak at this event. And when I first got the invitation, I was wondering what, I thought, if you wanted to live, age more slowly than others, I thought this problem had already been solved, through the special theory of relativity. So I was curious why they wanted me to speak on the subject, but I think we can give an alternative perhaps to having to move really fast if you want to age more slowly. So I've been trying to give this sort of analogy a few times now on how we perceive biology and how we can achieve understanding, not just knowledge, but get all the way to understanding, and thinking about living systems in terms of movies. So the same type of diversity we see in nature amongst wildly different species, some things in common, many things not in common. We see the same kind of diversity in a movie, and when you think about all that's going on when you're watching a film or a commercial, all the different information that's streaming at you is pretty phenomenal, and the ability of your mind to put all of these images together, all the symbols represented, their interaction over time, and you're able to consolidate all that information in a very, very short period of time and achieve understanding of what the clip or the movie is trying to convey to you. And some of this can be very subtle and very complicated, very nonlinear. And so when we think about biology and trying to solve the movie of biology, we can think about single dimensions and whether we can be successful that way. And of course, genetics has been one of the dimensions that's quite important, and the genetic study of disease, starting with Huntington's disease. Obviously, we all know a pretty horrific disease that Woody Guthrie, one of several who suffered from that, but able to bring genetics to bear and looking at families segregating this disease along with genetic markers, and with that, one of the first stunning successes of a genetic approach locking onto a disease-causing gene. Obviously, propagating a whole revolution that we see today still in full swing, from the whole exome to whole genome sequencing, and still bigger and bigger GWAS studies. But as far as achieving understanding, right, of the disease from that sort of discovery alone has been, somewhat limited. And I think with Huntington's, we see 30 years after the discovery, still no effective treatment or cure for that particular disorder, and we see this in other areas as well. So whether it's the ApoE gene for Alzheimer's and how much money has been dumped into that particular area and focused on that genetic finding and trying to get to novel therapeutics that are very effective or that can prevent the disease has been more of a challenge, I think, than many had thought. And of course, the reason for this to me has always been pretty clear, because if you try to look at this single dimension and try to understand this very nonlinear complex phenomena, you're not going to achieve understanding. And one way to see that is to think back to the short clip I showed you and all the information that's streaming by you. And now what I've done is I've taken each frame of that clip and I've simply averaged each pixel over all of the frames. And what you're looking at here is the average of all the pixels of that clip. And so you can try to look at that and say, what's going on in that clip? And you're not going to achieve any understanding or knowledge from that. And that's, again, the population average, single dimension, that's the kind of view that we had been taking to try to understand, something that's very complicated. And of course, it doesn't take much. Just, understanding a movie is not harder than understanding biology. And so we can see the limitations straight away. And so the idea is one of taking multiple streams of data, right, again, many different dimensions of data over time, and being able to build, these networks that are, again, integrated in highly nonlinear, complex ways. So here, just showing some of the key molecular dimensions, showing that they're connected in very nonlinear ways to form these networks. These networks define biological processes that carry out functions in cells and organs. And again, these networks don't act in isolation, right? They act in concert in a network of networks that define the complexity of living systems. And so this is really the view that I've been advocating now for a number of years, that we need to get away from the single gene view focus and into this more network-based focus, where once we understand some of the connectivity of the networks, we can start looking at how changes in one part of a network are propagating through to other parts of the network. Say this is within a given cell or tissue type. But of course, we're more complicated than a single cell or tissue type. We're made up of many different organs. And so the idea is, can we create these type of circuits, these sophisticated circuits representing the living system, where we're not only looking within a given cell or tissue, but we're seeing information propagate through the entire system. So it's one of embracing complexity, and the old Zerhouni, leader of the NIH back a decade plus ago, was pretty locked on to this idea and sort of beating this drum for a long time now, where, again, it's one of taking the sum of all the parts and sticking them together, viewing them holistically to get at the full view of what we're trying to do. And, of course, there are problems with embracing the complexity, and a lot of it has to do with orthodoxies, right? So we become accustomed to these long-held core beliefs that are no longer true, but they're held on to just for historical significance or just inertia. And the problem with these is they can really stifle, right, creativity and innovation. And of course, this isn't specific to biology. We see throughout human history. One of my favorites is Mr. Warner in 1927, talking about who's ever going to want to hear an actor talk in a movie. So those sorts are Mr. Watson of IBM talking about a world market for maybe 5 computers. So those are the kinds of orthodoxies that we sort of get stuck in, and then that can stifle our ability to be disruptive, creative, or innovative. So embracing complexity, I would argue, is difficult because a great philosopher long ago sort of told us this would be the case, where Francis Bacon, basically said that our minds are basically wired to think that more order exists than actually exists in chaotic nature. And so we see this again throughout. If you look in any textbook, even today, it's sort of surprising, in biochemistry, and you think about things like TGF-beta signaling, you'll be presented with this nice, linearly ordered pathway of how that signaling actually works. But again, the problem there is one of our minds being wired for storytelling, not for statistical uncertainty. And so these are the really simple constructs we like to convince ourselves are true, but in fact, I think know deep down, they're not true. So again, our mind is fighting us, and of course we see that throughout human history too. So it's not just specific to the biology or medicine or life sciences, biomedical sciences arena. It's throughout human history we see the want to tell stories about complex things we don't understand. So whether it's the Zeus, the sky god, throwing the bolts of lightning, or the Earth being the center of the universe, or the Earth being flat, or that biological processes are carried out by simple linearly ordered systems. These are the things that that can mislead us and cause a lack of progress. So ways we can fight this are through embracing of technology and new ways of thinking. And so I'll sort of touch on a few of these to launch into the network space. So the technology today to sense all aspects of life is just growing at an amazing pace, and we see this in many different areas of life. And some of the ones that are embracing this big data, the ability of sensors to generate big data and better inform on complex systems. The Oracle's Team Oracle big data America's Cup boat has 300 plus sensors outfitted on this boat that measure all different aspects of the operation of this boat, from the crew to all aspects of the boat, the sails, the hull, the ropes, and so on and so forth. And they're measuring 3,000 plus different variables, generating, operating at 10 hertz, generating about 300 gigabytes of data a day. And they're processing that data in real time every day to come up with better predictions or better tuning mechanisms for the operation of that boat. And they're actually dumping those servers overboard every day, every night, to be picked up by another boat that then does even more processing on the data for the next day. We see the same thing with Team Honda, the Indy 500. Again, hundreds of sensors on the car and the driver, monitoring all aspects of operation of the car and the driver. About 1 gigabyte of data is generated per lap, being, again, processed in real time, and adjustments are being made in real time to, again, optimize performance. What we see what do we see, though, in the great state of New York when a very complex machine, a new baby, is brought into the mix? Well, there we do a little prick on the heel and then smear the blood on a piece of paper, and then we mail that piece of paper, snail mail, up to Albany. And some tests, important tests get run, but it's a limited number of variables that are really informing on the health of that child. And if it's something super severe, metabolic rare disorder, that can get caught. But of course, if none of those things are flagged, the baby is sent on its merry way and won't re-enter that system unless something bad happens. So the question is, can we start embracing technology to build better models of human wellness through better sensing outside the medical arena, over time? And of course, this isn't something that most people here have not heard of. It's commercially, all the wearable devices, implantable devices that can exist now in your environment, aggregating lots of information. Some of these devices are pretty cool, like the sensing tattoos that are Bluetooth enabled and can measure all sorts of physiological parameters on yourself and pipe that information to a Bluetooth device. We have even babies, Pixie Scientific, with the sensors and the diapers to monitor things that are being excreted by your little guy, to maybe take some action there that you wouldn't have otherwise anticipated. And then pretty cool companies that are really tipping upside down the way we're going to better assay different parameters on ourselves. So Theranos, now operating in some Walgreens, has been very disruptive from the standpoint of using this microneedle technology to draw very, very small amounts of blood and then be able to carry out all the kinds of tests that you would normally get in a hospital where you have to have 10 mils of blood drawn in a formal setting. Here you can go into the Walgreens. The device, when it hits your finger, you don't even feel a prick or anything. It's amazing that it gets anything. And then you can just pick from the panel of tests that you want performed. And again, having that kind of access to any range of tests that you normally would have had to go into a medical setting to get, you can now get at Walgreens. And of course, there's other big data endeavors that I hear happen around this area with the NSA and others collecting big amounts of data in the environment. And some of this data does make its way into apps that can help one assess the environment that they're living in. So the Urban Science and Progress Center at NYU takes a lot of the data streams that are generated in New York City, looking at everything from traffic flow patterns to electricity usage to pollution levels and temperature and so on. All these different environmental contexts that can be very granularly assayed across the city are now potentially available for others to pull in to perform, again, better assessments on not only themselves but the environment in which they're exposed. And of course, it's all about then how do you aggregate all of that information and convey it to individuals in a way where they can actually make decisions, right? So and there are a hierarchy of users here, from consumers to physicians to researchers to payers and so on. So there are lots of different variables in the mix for how you're going to present that information in ways that people can actually act on it. So in the end, right, it's this big data universe we're living in, big data world. And our aim is one of how can we aggregate all that information, build these predictive models that can better impact our ability to diagnose and treat disease. And it doesn't have to just be happening in the medical arena. This could be stuff that's running on a smartphone for the consumer to help them make better decisions on their own health through better analytics off the data that's being collected on themselves and in the populations that they're part of. So one of our goals has always been how to make sure the molecular dimensions of data are in that mix and try to link the molecular biology up to the physiology of the system. So we can collect all this physiologic sensing stuff going on in the individual, and of course we want to link that up to all the molecular business, whether it's DNA information or metabolites or proteomics or RNA. We want to be able to integrate that information together to get to these predictive models. So how we engage the information, again, is going to matter. So the one addressing how we're going to engage complexity is through the embracing of the technology and leveraging technology to aggregate more information. But then how you view that information is also going to matter. And this was a neat behavioral experiment. Many of you may know much better than I, but I thought this was kind of cool in showing how if you present the exact same information to people in slightly different ways, how it can impact their ability to solve the problem. In this case, the problem was to affix the candle to the side of the wall, light the candle without wax dripping on the wall or the table. So that was the problem. And the two groups that were asked to solve the problem were given the exact same material to solve the problem, but presented it in slightly different ways. In this case, the tacks were inside of the box, and in this case, the tacks were outside of the box. And on average, group 2, the group where the tacks were outside of the box, came up with a solution twice as fast as the group 1. So just by presenting that information in a slightly different way, you had a pretty dramatic increase in the rate of solving that kind of a problem. And so the key here to me was one of, as we become more expert, as the grooves in our brain become deeper, that we become more blinded, right? We have the blinders on because we're trained in a specific way, so we have less of an ability to think outside the box and to be disruptive. And so you have to be really conscientious about that and set up the structure of how you operate in ways that enable you to not be blinded as the expert. And again, we can take lessons from others on this. This isn't, again, unique to the biology field, and here's just a few examples of how that's been done. And one of my favorites is with BMW, because I'm a little bit of a speed freak and I like to drive my S1000RR and my M5 only in very safe conditions on tracks at high rates of speed. But what BMW did is they sat engineers with people like me thinking that, oh, these guys who buy BMWs, they're all about performance, and we want to know how do we better tune that car to accelerate better, corner better, brake better, and so on. So they drove around with the customers, and they were shocked to learn that the number one feature that was talked about from the customers of BMW had nothing to do with performance, but was their concern about pulling into and out of parking spaces and the damage that can be caused to their car as a result. So pretty interesting that the park assist features came to be as a result of that. So BMW pioneered that. But again, that was something that wasn't, that any of the experts before sitting with the customers would not have predicted that that's what they needed to do to go to the next level, that it was only through engaging the information the consumer was giving in a more direct fashion. So in our case, what we're going to do is engage the information in a more aggregated way. So I'll start with sort of the traditional way of how we might figure out variables of interest that may be causing disease or adverse events to drugs and so on. So we have two variables, and we're looking at these variables and sampling through a population, and we see that they're correlated over time. And based on this correlation, we might come up with a hypothesis that says, okay, we think this gene is affecting this clinical feature in a causal way. And so then you go into the lab, and now you're looking at a given model system over time, and so you set up this experimental system, and you're looking at the variables of interest, and now you're going to carry out an artificial perturbation on the thing you're predicting as causal for the other trait. You're going to set up that system, you're going to do the perturbation, see the response, look for the subsequent response in the other variable, and based on that chain of events, establish a causal relationship. So that's one way to do it. That's been obviously hugely successful, lots of amazing discoveries made in this way. But another way to do it is to leverage and aggregate more of the information that's perturbing biological systems in a more systematic way to get at the kind of networks I showed you in the animation. And the way this kind of works, well, maybe I'll show you first. The idea is one of taking DNA variation and looking at the DNA variation and its correlation to these traits of interest, and then stratifying. This is not wanting to work. Well, so what it should do is stratify this population into three groups, right? So you take a biallelic SNP, you have three possible genotypes, and you can split the population into three different groups. And what you're doing, think of a clinical trial. Like, how do you establish causality in a clinical trial? Well, you have two arms. You have a placebo arm and you have a treatment arm. And what enables you to infer causality in giving the treatment to the individuals, the randomization of patients to the different arms of the trial. So it's the randomization mechanism that gives you the ability to infer the causality. Well, this is the same sort of game that we can play in the genetic space, because if, say, you have a biallelic marker variant, say it's causing things to happen, well, which version of that allele you get is randomized during meiotic divisions, right? So it's that Mendelian randomization concept that we pioneered, it's been back in 2003, that gives you the ability to make causal inferences based on those genetic perturbagens. So again, the setup is DNA variation correlating with two phenotypes of interest. And if this is the case, because the variation in DNA is a causal agent, there's only one of three ways these variables can be related. Either the variation in the DNA causes phenotype 1 to change and subsequently phenotype 2, or vice versa, or that variant independently affects those. So we can basically come up with a mathematical way of modeling this, which I've been doing for the last 10 years, and we can apply this systematically throughout the variable space to come up with a network. So again, here we have all the different nodes, could be hundreds of thousands or millions of nodes, that if they're impacted by DNA variation, we can apply some mathematics and then statistically infer causal relationships between them. We can do this in a very systematic fashion over the entire variable space, and from that come up with these causal probabilistic models that are actually predictive of the different traits that we're interested in that are associated with the population that we build these networks from. So whether it's a disease phenotype or a wellness phenotype or a drug response phenotype, any of these things can be applied in this sort of framework. In addition to DNA variation as a systematic perturbation source, and again, just to, the importance of DNA variation as a perturbation source is it's the variation in DNA that's causing these different diseases, at least partially, or response to drugs and so on. So it's a relevant variation source off of which we should be building the networks, but we can also employ time-based causal reasoning and actually integrate both the time and the genetic information to come up with a more holistic causal network framework, and these are things that we now do routinely. And the ultimate goal is one of getting at complete computational models for a living system. So just like in engineering, where you now build, whether it's a sophisticated jet aircraft or a space aircraft or whatever, that those are no longer built initially, physically. They're built on the computer, and you go through simulations, and they figure out exactly how to design it and so on. And then they, once they're confident in the design in silico, they go to make the actual machine. Well, we want to be able to do the same sort of game in biology. And one of the guys who recently joined my group, Jonathan Carr, had a paper in Cell a year or two ago on the first fully computational model of a living system, where it was blanking on the bacteria now, but it was maybe 600 genes in the bug, and he modeled every aspect of the biology of that system, from the DNA synthesis, DNA replication, to the transcription, to the translation, to the metabolic flux from both the environment and then within the system. So he took all of those different aspects of the system and computationally modeled them and strung all those models together to come up with a holistic model of the fully computational model of the system. And then he's able to do biology in silico, right, by perturbing the different elements in that model as opposed to doing it in the lab. And so here's just an animation that he had where here you're looking at different cells that are being simulated. You're looking at them over time, and the thing you're looking at is the growth rate of the cell and ultimately cell division. And all the different parameters of this cell are being modeled, again, from the replication of the DNA to the transcription to the metabolic flux to the protein translation and so on, all being monitored in real time. And the cell that you see changing there, that's being driven by all the underlying modeling that's going on. And so what he was able to do was show that he could go in and systematically perturb all the genes, either singly or in combination in that bug in silico, and came up with completely novel insights on cell growth and cell division that hadn't been anticipated, that were then validated experimentally. But again, I think one of the futures coming, obviously this is a pretty simple bug and we're a little bit more complicated, but again, the scales of data, the diversity of data, and the computational know-how are now sort of coming together to be able to carry out this kind of game. So my idea is one of, that our biology of yesterday was more about single genes, transcripts, proteins, and phenotypes, say a given disease, to today where technology has really given us the ability to accurately assay over, really the whole system, whether it's genomes, transcriptomes, proteomes, or phenomes. And so that's, but what we have to be applying to that is the right analytics, computational know-how to get to these causal network models that I think will be a key driver in the biology and medicine of tomorrow. And so now I'll just step through a few examples of where we've applied this in the real world setting and are uncovering some pretty interesting things. So this is a network. I didn't get a slide to preface this by where this came from, but this was a network that we derived from a study that was carried out by, of all people, Deepak Chopra. So I don't know if you know Deepak Chopra, but he's a meditation guru guy. And I got interested in that because what we saw in the placebo arms of trials is when we looked in the placebo arm where they have molecular data and we looked at responders and non-responders in the placebo arm of trials, we saw very significant molecular differences between them that were mapping to really interesting networks, and I wanted to see whether those same networks were at play in response to meditation. So what this network represents are people who went to, and this was a study carried out with Alyssa Apple, Rudy Tanzi, and Elizabeth Blackburn, who many of you will know, again, better than I, where the trial with Deepak was one of people came in to this meditation week-long thing. They tried to, they were asking me if I wanted to do it, and I said, well, I'm way too ADD to be sitting there for a week just meditating, and that would drive me just completely crazy, and that instead I'm going to wait for the pill that obviates the need to do that kind of meditation. But the people were randomized to either a vacation arm where they came to the resort and all the amenities of the resort, they could relax and do vacation, or the meditation arm where they actually went through this intense meditation, and then blood was drawn pre and post, and then we RNA sequenced that in addition to other biochemical assays like that Elizabeth Blackburn carried out, like the telomerase activities pre and post and so on. And so this network is built from all that data using the techniques I just described. But what was interesting about that is there was a clear difference in the molecular processes that were being affected from relaxation, just being on vacation, that were very positive, but also molecular features that were being affected purely in the meditation group. And so what you're looking at here is the green edges, the little blobs are subnetworks of this bigger network that were enriched for genes that were either specific to the vacation effect, so those are genes that are involved in the green edges, or specific to the meditation effect, and those are edges that are dominated by or that are encoded red. And so you can see some of the subnetworks that were very coherent, very interconnected, being specific more to the meditation-specific effects versus the vacation-specific effects. And so we looked at some of these in more detail and was sort of astonished to see that in this big study, so I can't say what the genes are because Liz is still publishing on the GWAS for the telomerase activity and telomere length thing, but this particular network was over 10-fold enriched for genes that were discovered through the GWAS or have known rare variants that cause telomere-related disorders, 10-fold enriched for falling in this network that was specific to the vacation effect. And then we had others, and then this particular network was enriched for things like response to stress, defense response, inflammatory response, wound healing. And what you're looking at here now is the same network of genes that were different between pre and post vacation. And the green, the coloring is the levels of expression relative to the baseline. And so what you're seeing in this is green is down, red is up. And so you're seeing systematic downregulation of this network that is downregulation of all these defense, immune response activities that the system would normally carry out in your normal day. So kind of making you, so the relaxed phenotype sort of coinciding with your body relaxing and going into a less defensive posture. But then equally interesting was this particular network that was very specific to the meditation and very enriched for genes involved in mitochondrial function and very enriched for genes that are involved in, when disrupted, lead to all sorts of mitochondrial diseases. So being able to partition that sort of thing apart is hopefully going to help us better understand how even the human system, through better behavior, maybe we don't have to meditate, but certainly relaxing and maybe going into other sorts of behavior modification may help us live longer, healthier, better, well lives. And I think bringing this type of rigor to those kinds of studies can help. And we have a couple of ongoing studies I can talk about afterwards if you're interested. So that's one end of the way out there on the behavior modification spectrum, but we also have work going on more on the mechanism side of aging, where we're in this case looking at yeast. This is in collaboration with John Hartman, who leads a good group at University of Alabama, where he uses a yeast model for aging. And again, just the yeast is a good model system to start exploring some of the core mechanistic underpinnings of things like aging, not a completely out there idea. In this particular case, there are a couple of different models that are looked at from the standpoint of yeast from aging, these replicative lifespan models and the chronological lifespan models. We're mainly focused on the chronological lifespan models, where we're looking at basically these different populations of yeast, again, using genetic variation as our systematic perturbation source, looking at these wildly different naturally occurring yeast strains published in Nature a few years ago, but basically profiling all these guys with respect to a very unique image-based growth assay that John has developed to look at longevity of the yeast in a highly controlled environment. And so this just giving you some early example of here are the cultures. You're looking at the growth of these cultures over time. And you can see how these different strains, here are just 3 different strains cultured, and you can see that they grow very similarly when they're young, but the longevity of these guys is highly variable given the different genetic backgrounds, as you can see. So these are, as the cultures are getting older, they're dying out at different ages, some living very long and some dying off very short. And so we've already identified here, just showing you some of the different growths of the most varying strains. This pair here living a long time, this particular species dying off much more quickly, and then carrying out crosses between those guys to play the same kind of game we would play, whether it was Alzheimer's or obesity or diabetes. We can play here to understand what are the key nodes that are driving the variation in the aging. And so we've just now started getting a handle on the networks that are involved in this particular process. So I can't go much further today on than just some of the early pictures of the networks for the genes that are involved in explaining the variations in the ages of these guys or the longevity of these yeast. But as we do the crosses and are able to more formally layer in the genetics of this, we'll get at the causal underpinnings, at least in that organism, and then can map that back to human. So those are sort of two ends of the spectrum, but where we're spending most of our time are on diseases such as Alzheimer's, obviously pretty horrible disease that's of high public health concern to many in this room, more expert than I am in this area. But the idea being, can we employ these same sorts of technologies to get a better handle on Alzheimer's disease and to better diagnose, treat, and prevent this disease, which is obviously going to cause already causes lots of havoc today, but as the population ages, it's going to become a much bigger problem. One of the things we put a bit of thought into is, if you're going to study Alzheimer's, what part of the brain do you really want to go after? And there's, of course, the parts of the brain that that are most commonly associated with the pathology of the disease. But we decided to take a more data-driven view just to complement what other groups are doing. So we had from the Mount Sinai Brain Bank 20 or so different regions of the brain where 60 or so individuals were profiled. Again, these were common amongst all the individuals that were profiled. All these different regions were profiled. And the idea was, can we lock onto, so these were all individuals with Alzheimer's, and we wanted to see whether we could take a more data-driven approach to identify what are of the 4 regions we're funded to pursue, what are the regions we should pursue. So we're looking at, first of all, each of these plots are showing the degree of correlation of the expression traits within the different regions that we had profiled. So each bar represents a different region of the brain. And then we're looking at the degree of correlation of the expression traits in those different regions of the brain to the different clinical features that were scored in this population. So things like the Brock score and CDR and so on. So looking at the degree, what regions of the brain are capturing the most variation in these clinical features was one thing we were interested in. The other thing we were interested in were differential genes that were not only correlated with the clinical features, but were differentially regulated between the different tissues that were being profiled. And so this is just a plot that's trying to show you the degree of enrichment for differential expression across different combinations of tissue that we're looking at. So it starts off in this radial plot of comparing all pairs of tissues, and then you go into all 3, N choose 3, and then N choose 4, up to, we just are showing up to 6 here because the number of genes that are intersecting basically goes down to 0. But basically using this differential expression, like what of the expression activity in these different regions are able to best explain the clinical features and be differentiated between the different tissues. And then we're layering in the networks as well, the crosstalk between, remember the animation I showed, and we're interested in the crosstalk, not just between the organs, but within an organ as complex as the brain. We're trying to look at all the different features that are interacting in the brain and looking for regions that are more highly connected across different parts of the brain. So not so completely isolated, but being connected to all the different regions of the brain. And again, we can derive all of this from the molecular information in those data that we generated. And so surprisingly, we came up with basically a ranking score across these 20 regions that were based on the degree of association to clinical features, the differential regulation of those correlated genes between the different regions, and the connectivity of the different regions of a given region to the different regions. So we came up with a machine learning based score where we rank ordered all of the regions and came up with these top 4 as the ones that were most relevant to study. And the interesting part about this is that these weren't maybe one of these regions was one, if you asked the experts in the room, that they would have chosen to profile, but the other ones, were sort of never would have thought to do that one. And of course, we're still in the process of generating the data, so hopefully we don't get humiliated down the road that those were the wrong four to go after. But again, the motivation for this was one of taking a very data-driven approach by letting the data speak to us and tell us which ones were looking the most relevant to that disease. So once we have that sort of information, another region we did just because we wanted to be in common with all the other groups who are generating this data is look at prefrontal cortex. So this is work we had published where we looked at 500 prefrontal cortex samples, half from individuals with late onset Alzheimer's, the other half non-demented controls matched by age and sex. And so we can build all the networks that I've been talking about through the generation of that data. So we profiled the RNA on those data. We did the DNA genotyping on those guys. We had a bunch of other biochemical tests and the histopathology and so on available on these individuals. And one of the things we noticed about these networks that I thought was worth highlighting is that this idea of differential connectivity. So because we had disease and healthy, we could construct two different networks for each and then compare those two networks to say, are they connected in the same kind of way? The surprising thing was that the differential expression between the disease brain network and the normal non-demented control brain network is there were very few genes that were differentially expressed in prefrontal cortex for that, which was very surprising given that's the tissue that's being torn down. So what we looked at in complement to that was this differential connectivity. So what you're looking at here are all the different sub-networks that we identified, the different colored networks that I showed in the animation. And what we're looking at on the upper triangle are the genes and their degree of interaction in the late onset Alzheimer's. On the lower triangle, it's the normal. So the more red you are, the more interconnected and correlated the genes are. The whiter you are, the less connected, the more diffuse you are. And what you see straight away are very striking patterns of differences in connectivity between the Alzheimer's brains and the non-demented control brains. So even though the differential expression was not that great of a signature, the differential connectivity signature was enormous. So again, just highlighting that how you look at the data, the type of metrics you use, and the type of more holistic views you take that you get from the network connectivity can lead to very dramatically different results. And so here we have one of the, this is the network depicted as a whole, and one of the highlighted networks was one of the most differentially connected networks in the brains of individuals with Alzheimer's versus those without Alzheimer's. So that's being highlighted here. So in this case, all the pink and yellow nodes are either gene expression traits or DNA loci that are correlated with DNA expression traits or clinical features. And the blue and red edges are all the causal links that we're inferring between these guys. And then I've simply projected one of those subnetworks that was most differentially connected onto this network, and that's what's highlighted in the yellow and red. So this is an area that's very disrupted in the Alzheimer's brain versus the normal. And if we look at this guy, we can see lots of different, so this is just zooming in to those yellow nodes, and the red edges are now depicted here. And you can see lots of different pathways that are interacting here. And the interesting thing about this was all immune inflammation related. And so a lot of these pathways we had seen in other diseases. So when we compared this particular network across many different diseases, we found that the same network was underlying partially a whole range of diseases. So for example, we took animal models for atherosclerosis, diabetes, inflammatory pain, COPD, asthma, fibrosis, and so on. So 12 different diseases where we looked at the disease tissue versus a wild-type normal tissue, did the expression profiling, looked for genes that were differentially expressed between those tissues, and then filtered based on the genes that were in this network that we identified for Alzheimer's. So what you're seeing plotted here is a color map where along the columns are the different genes in that network, and the color coding is the differential expression activity between the disease and the healthy tissue, and you can see a high degree of similarity across those different diseases, that this network is oriented in the same kind of way across a whole range of diseases. In fact, when I projected on the same network, I looked at the Alzheimer's loci So these are highlighted in orange nodes. So you can see ApoE, TREM1, TREM2, CD33, and so on. So this network that I just showed you is tenfold enriched for loci that are being discovered in the GWAS for Alzheimer's, are enriched in this network. The pink nodes, though, are genes that are implicated or genome-wide significant for inflammatory bowel disease, where this network is 20-fold enriched for genes that harbor variants that cause IBD in the human population. So really interesting just seeing this integration of all this information and genes that are causing IBD also connected to genes that are causing Alzheimer's, even though these are occurring in completely different parts of the system. And so when we, what do we want to do then with this information is we want to now start doing biology in silico. We now want to take this probabilistic causal structure and start perturbing all the genes to understand which is the gene we should go after for therapeutic intervention. So the way we do that is we systematically go through and hit every single gene in silico. We knock it down, we overexpress it, and then we see what the model predicts the response is going to be. So here you see you hit that gene, and it's only affecting this one other gene. We hit this gene, it's affecting two other genes. But sometimes we encounter a gene that systematically affects the entire state of the network, what we call a causal regulator. And those become very interesting to us because they're modulating the entire state of the network. So when you start thinking of biomarker or therapeutic intervention, these are the nodes that we care a lot about. And this is just looking at the structure of that network in a slightly different way, where now we've organized all the nodes in the network by key drivers that are listed here in the bigger nodes, and all the downstream, more proximal downstream targets, and the more even further downstream targets of those key drivers. So now we can start really using the structure of the network to actually lock onto what are the things, where are the places we actually want to place the bets for this. So we identified through this novel gene that was modulating this was a key driver, TYROBP, otherwise known as DAP12. We identified as a key driver for this, modulating the state of this network. It signals through TREM2, so it was interesting that our study came out around the same time as the TREM2 discovery, but basically able to identify what of those nodes in the network we should want to think about targeting. We went on and validated that discovery in vivo. And the next step we want to do, and this is almost just a couple of slides left on how we can actually get to a therapeutic intervention sooner rather than starting from scratch in a drug discovery program, is we can map all of the existing drugs, so natural products, existing FDA-approved drugs on the market, and we can map them to the nodes in the network that I was just showing you. So now the blue nodes here represent the network that I was just showing you for Alzheimer's. That's also at play in all these other diseases. And the green nodes are the drugs, known marketed drugs or natural products that are known or predicted to interact with the coding products of these blue nodes. And the pink nodes are disease-associated traits or disease indicators that are known to respond to different genes in that particular network. And what you should note straight away from this is that's a pretty complicated array of information, of interactions going on with different drugs and different genes and different clinical features. So the idea that we're going to pick one drug and we're going to orient this network in the right way to treat disease is just not true. So what we're thinking about is how do we pick constellations of those guys to orient the network in the right direction to ultimately have a positive effect, not just on the disease indication of interest, but on all the other diseases that may be affected by moving that network around. So think of adverse events and other things that we would like to avoid. And so we carried this out on this particular, the network I was showing, where we identified SP1. So this is a gene that's localized in a network that was heavily associated with Alzheimer's, and that we then screened against the connectivity map data to see if we could find drugs that were matched to that particular signal, and found a whole list of drugs. Unfortunately, I'm not at liberty to say because it wasn't all my work. This is work being led by Joel Dudley in my department. But came up with a list of drugs that was heavily impacting the state of this network. One of them, this tolfenamic acid, Yeah, so I don't know how to say it, but you guys can see it. And this particular guy was published not too long ago, demonstrating improved cognitive function in Alzheimer's disease in mice. So it was sort of a nice, positive hit for us that was already been validated, and we now have in testing in animal models the other top drugs that hopefully will have an impact. But again, remember, these are either natural products or existing drugs that could go very rapidly into a human proof of concept study. So we wouldn't have to go through the whole rigmarole of all the safety trials and so on, that there's the potential to, using biomarkers generated from the network to give the drug and see whether we see movement in the biomarkers we predicted. And if we did, then maybe that becomes something that you're willing to place a bigger bet on to run a more formal trial. And at the end of the day, what we hope comes from all of this is a better understanding of human health, that all of these network models, that we can interpret all the perturbations that occur in individuals. This is the precision medicine game, right? What are the perturbations that are happening in your DNA, in your environment, that are disrupting the molecular networks that underlie all the processes that cause disease? So we want to be able to match individuals to the networks that are being disrupted in them. And then based on that, just like I showed you for the Alzheimer's drug, can we match those networks to the most appropriate treatment or the most appropriate behavior modification, whether it's diet or exercise? That's the kind of, what we ultimately want to wrap around all of these models in an actual clinically actionable way. So we're actually able to take an individual, all the information generated on them, make these sorts of matches, and then be prescribing things based on that type of information. And we have a number of studies ongoing in the cancer arena that are playing this game directly. So with that, I wanted to thank you for your attention, and obviously many collaborators involved. The group at Mount Sinai growing bigger and bigger, a very fun group to be interacting with, but many other academic and industry collaborators that have been involved with this work. So thank you.
So those with questions want to step up to microphones?
Eric, over here. So it's a terrific talk, but I have a couple of questions. You're drawing causal maps, but you're giving us an indication that they're really molecular maps because you're targeting them with particular drugs and so forth. But you're taking whole tissue that's composed of many different cells that have different expression of certain molecular components, and some are present in some and some are present in others. So how are you deconvolving what's going on in individual cells, which are really the networks you'll affect with a drug, as opposed to the overall network in a complex tissue that have glial cells, neurons, and so forth, all mixed in together, for example, in the Alzheimer's case?
Yeah, so it's a good question, and I think for a couple of things. First of all, the networks that we build from the whole tissues, they do deconvolve into these very well-defined subnetworks, these modules that at the grossest level, the topmost level, are highly reflective of cell type. So even though you're profiling something that's very heterogeneous and potentially complicated, what those cells are doing, there's a lot of unique features of those that stand out clearly in the network that you can model. So we think that we do get at cell type specific. The network I was showing you, right, that came from brain, yet it reflects almost perfectly what's happening in microglial cells. So even though there are neurons and astrocytes and other things going on in the mix, that particular network was nearly 100% enriched for genes that are specific to macrophage-like function, so microglial cells. The second point I'll make is that what we're seeing more and more, and this is playing out particularly in the cancer arena, is that a system's ability to respond to a drug is context dependent, and it's not just a single cell that's responsible for that. It's the entire system, and in the cancer space we see clearly the immune system. So even though things are targeting the drug, the state of the immune system can have a really big role for how you're going to respond to that. So I think there's a lot of advantage to having the more holistic mix of things going on to interpret that. So I think those are some advantages, but I'm also getting you too that if we could go down to single cell level and cell sort and be sequencing individual cells or individual groups of cells of a particular type, like we would love that, but unfortunately the state of the technology to do that on a massive population scale just isn't there. So I think we're trying to deal with the best that we have.
The other question is, it may not be relevant at the level of therapeutic intervention late in a disease, but the difference between what is causing the disease to begin with and what the reaction to the tissue damage is, which is almost always going to be these macrophage signatures you see in all of these different conditions, are two sort of separate questions in terms of thinking about prospective treatments as opposed to just therapeutics. And so are you beginning to parse that out? Because clearly this common signature could be a driver in that it's causing everything, but it could also be a response to primary tissue damage that then perpetuates or accentuates the disease. And that might be a good therapeutic target anyway, but it also may not cure disease.
Yeah, I love it. And I think it is one of the pressing problems that we're trying to address in the GWAS and applying this approach is, can we start distinguishing the primary causal genetic lesions from the more secondary ones? So we do think that targeting the networks I was showing you, even though they are immune inflammation related, are going to have a efficacious benefit, because we see that those genes, genetic mutations in those genes, are strongly associated with this ApoE is a great example of a number one risk factor for Alzheimer's occurring in that network. TREM2 is another one, CD33, and so on. So I think it's highly relevant as implicated by the human genetic data, which is causal. But yeah, you're absolutely right. The ability to start teasing apart those mutations that are earlier on in the disease and actually, causing, like the plaques, say, versus the immune system responding to those plaques, I think is something that we are confident that the network will be able to shed some light on, and it's a very active area of research for us. It's a great point.
Since you were quoting Francis Bacon, I have a philosophical question for you. There's a huge chasm between correlation and causation. Correlation we can observe, causation we can only infer. And so my question is there more here than a multiple correlation? And anticipating that the answer is yes, can you cook down to some simple principles what is allowing you to bridge between correlation and causation here? Yeah, so
definitely a good point. And I grew up through this mathematics and statistical arena, so well aware of this correlation causation debate. But what we have in this case, so what do you need to resolve a causal relationship between variables that are correlated, is you need a systematic source of perturbation. You need a perturbogen. In classic biology, you have two things that are correlated, and you'll directly perturb that gene. You'll knock it down, you'll overexpress it, you'll chemically perturb it. Whatever your choice, CRISPR-Cas, whatever your choice is, you can directly perturb it, and then based on that perturbation, you can make that causal connection. So what I was advocating from the DNA variation standpoint is the DNA variation provides the exact same avenue as the artificial perturbation, but it's naturally occurring. So a DNA variant that's knocking a gene down or overexpressing it is just as valid a perturbation, if not more valid, than what you would do in the lab to make that causal inference. And so by leveraging that as the perturbogen, we can play the same game you're playing in the lab to make the causal inference.
Yeah, just a question we talked about a while ago, and it relates somewhat, I think, to Ron Germain's question. So when you talked about nobody building an airplane now until it's done in silico, one can take the question now of how when you identify these networks, they make a prediction about what might be a favorable intervention when we're talking about translation. How can you adequately model from a multicellular circumstance when you don't know at what point in the process, through what cell interactions you're intervening, short of going into the human? So do you have thought as well in terms of links and collaborations with synthetic models to stand between the in silico model and ultimate application to a whole organism or human?
Yeah, it's another great point. And obviously when I am talking about this stuff, I'm excited and on the edge of, selling it. But it's clearly a long way to go on the network-based models to get at stuff that's truly accurate, where we can build, the plane without, looking at, do that in silico before we do it and for real. So I think there's a couple of different ways we're approaching this to get at Rich's point. One is we're trying to unify structure-based learning with bottom-up sort of modeling. So we're trying to take, like, what are the things that can be done in vitro systems or organoid systems, or like the Weiss Institute and the more complicated organoid systems are building that reflect more of the different organs in the system. Like, basically, how can we integrate that more mechanistic information? So if you think about the models, there's a whole spectrum from the dynamical systems, differential equation type models, modeling just a few variables where you think the pathway is exactly known, the relationship is known, and you're just modeling the flow of information, all the way to the structure-based learning type stuff that I talk about, and how we can unify those two mathematically to make better use of the type of data that's being generated in these sophisticated in vitro systems, or the kinds of experiments that are very mechanistic based and very focused on individual pathways or genes. Like, we want to be able to seamlessly incorporate those simultaneously. So there's a lot of work going on to make use of all of that kind of information in a more holistic mathematical framework. But a long ways to go, I think.
So with apologies to the questions remaining, at this hour we'll close the formal session with thanks, but please feel free to come up, and let me also remind everyone that thanks to the generosity of FAES, who is sponsoring a reception just outside and down the hall in the library, another chance to be speaking with our guest for today. Thank you so much.